Hello, in this video we will discuss about the photosynthesis and the production of glucose and starch. And let's begin to understand with the whole steps and whole processes. So here is the autotrophs. This is the photosynthetic plant. It is autotrophs, it produce energy. So carbon dioxide fixation, water fixation and lead to produce in the presence of light and chlorophyll to produce the glucose. So this is the cell of plant which that's use the photosynthesis. In this way, here is the nucleus, vacuole, mitochondria, and chloroplast. Chloroplast is very important organelle, and this is the cell wall. Chloroplast is used for the photosynthesis and produce glucose. So let's begin to understand first the structure of chloroplast, and we will discuss also next electron transport chain for production of ATP and NADPH for using in the Calvin cycle in the dark reaction. So this is the light will be fall on the thylakoid membrane. This is the double membrane organelle, and this is the lumen inside the thylakoid membrane, and this is the granum. And outer membrane here is an inner membrane is the grana and contain granum. And in this way thylakoid membrane here, and here is the stroma for dark reaction. Stroma is used for the dark reaction because the rubisco are present in the stroma. So this is the chloroplast photosynthetic pigment containing organelle is used for the photosynthesis for production of production of glucose. So here I am not drawing the structural formula and the equation of the photosynthesis, but you can make sense about this. So here is the electron transport chain. It is the light reaction, and light fall on the thylakoid membrane to produce ATP and NADPH. Let's begin to understand how it's possible. So here is the light will be fall on the photosystem 2, which that's contain thylakoid membrane. Its photosystem 2 contain a many complexes. Basically it's not complexes, but it is the antenna complex. So antenna complex which that's contain primary electron acceptor photosystem 2 contain photon 680 nanometer absorption of light containing pigment. So in this way here is the lumen and this is the stroma. So this is the thylakoid membrane. So this is the oxygen evolving complex. This oxygen evolving complex means the water will be used the photolysis to produce oxygen, proton, electron. So in this way here you can see this after the production of through water, 2 molecule of water will produce the 1 molecule of oxygen, 4 proton and 4 electron. But in this way we will get a sense with the 1 electron transport for understanding just. So here is this is the photosystem 2 which that transmits the electron to the plastoquanine. This plastoquanine is basically reduced with the accepting of the proton and electron to form a pKOH2. This pKOH2 will pump in the proton more. It means the inside the proton will be more and outside in the stroma will be proton less. So acidity will be increased in the thylakoid membrane lumen. So here is the cytochrome. The cytochrome also next acceptor of electron will remove the proton and accept B6F. This is the B6F is the cytochrome which that accept the electron. After this accepting of the electron, the plastocyanine will be mobile protein will accept this electron to transmit this electron into the photosystem 1. Same mechanism, but it's contain primary electron acceptor containing photon 700 nanometer absorbing light to excite this electron to the next protein, ferridoxine. This ferridoxine is the iron containing protein. This iron containing protein will give the electron. This is also mobile protein, and in this way it will be transmit the electron to the ferridoxine NADP reductase enzyme. This NADP reductase enzyme, NADP plus reductase enzyme, to reduce the NADP plus into the NADPH. But where is the proton? So the proton is come from the stroma, not from the lumen. So it means lumen contain higher number of proton will pump out through ATP synthetase enzyme. It is the channel-like structure contain F0, F and F1 particle. So this is the proton, 4 proton required to produce when pump out this proton due to the proton gradient will be produced to produce ATP. And this ATP and NADPH will be used for the dark reaction into the stroma in the Calvin cycle. So what is Calvin cycle? Let's begin to understand. This is the dark reaction. So this is, for example, here I am again drawing for understanding the photosystem. So when light will be fall, it means the light reaction, but when absorb the carbon dioxide, it means the dark reaction. For fixation of carbon dioxide in the stroma, in the presence of Rubisco enzyme in the stroma, and here is the light independent reaction is known as Calvin cycle, because it is discovered due to the Melvin Calvin. So here is the carbon dioxide will be fixed. In this way, the fixation of carbon dioxide due to the ribulose 1,5 bisphosphate, 5 carbon containing ribulose phosphate, which that is bisphosphate, 3 RUBP molecule. This 3 RUBP molecule will be fixed 3 more carbon to produce 18 carbon. It means the 18 carbon means here is a carbon fixation step. So this is the Calvin cycle carbon fixation step to produce a 6 molecule of 3 PGA, 3 phosphoglycerate. These 3 phosphoglycerate contain phosphate group, and in this way the 3 glycerate, because the removing of the proton, absence of proton is known as glycerate, will use the 6 ATP and 6 NADP H to produce 6 NADP plus and 6 ADP. This is the stage reduction, second stage. And in this way it is occurring in the stroma. Here as you can see, this is the chloroplast and this is the thylakoid membrane, but this is the stroma. Stroma is used for this dark reaction because the Rubisco enzyme is present, which that will be fix this and work throughout the cycle. And in this way here you can see this is converting after the reduction of this molecule to produce 6 glyceraldehyde 3 phosphate, that is the sugar molecule, 6 molecule, but the one will be removed. In this way here is the stage reduction. In this way 3 molecule glyceraldehyde 3 phosphate, 1 molecule will be separate and 5 will be left behind, and in this way the 3 carbon will be removed from, and again the 5 carbon containing RUBP will be formed, and in this way the regeneration requires 3 ATP. After this, gluconeogenesis will be occur for production of glucose. Let's begin to understand. So this is a G3P glyceraldehyde 3 phosphate will be in the presence of enzyme triose phosphate isomerase enzyme to produce dihydroxyacetone phosphate. This is the 2 molecule, because the 2 running of the Calvin cycle will produce 2 molecule of G3P will be used, and here is the aldolase enzyme. 2 dihydroxyacetone phosphate will be used by the aldolase enzyme to produce fructose 1,6 bisphosphate, 2 phosphate containing fructose molecule, and in this way the fructose 6 phosphate formation, because the fructose 1,6 bisphosphatase enzyme, which that will be separate the 1 phosphate group, and in this way the 1 phosphate group will be separate, and phosphohexose isomerase enzyme lead to produce glucose 6 phosphate because the isomerization. So the glucose 6 phosphate will be formed, and this glucose 6 phosphate is ultimately converting through a glucose 6 phosphatase enzyme to produce again glucose after the removing of the phosphate group, to produce sucrose after the combining with the fructose. So this fructose will be combined here to produce sucrose, but the starch synthase enzyme are present in the amyloplast to produce the amylopectin and starch, amylopectin and amylose. So let's begin to understand with the biosynthesis of starch. That is very important topic, because the starch, where is the starch and how it's starch produced. So remember, starch is insoluble in water, so that is why the less requirement of the water is very important and crucial mechanism for the conversion of glucose into the starch, because the glucose is more soluble in water. So this is the cytoplasm of the cell. Fructose 1,6 bisphosphate will be converted through a fructose bisphosphatase 1 enzyme to produce fructose 6 phosphate. because one phosphate group will be removed by a hydrolysis, and in this way the phosphoglucose isomerase enzyme used to produce fructose as well as glucose 6 phosphate. This is the two step, and it can be fructose form. So here is the glucose 6 phosphate in the presence of fructose, glucose mutase 1 and 2 enzyme used to produce the mutation will lead to glucose 1 phosphate formation mutation. So it means mutation will be occur. So here is the important thing is that the glucose 1 phosphate will be lead to UGP1 and UTP will be separate the pyrophosphate, and in this way the combining to form a UDP D glucose, and in this way UDP glucose, and in this way the fructose used for separating of the UDP, and here is the fructose 6 phosphate will be formed and sucrose formed through a hydrolysis will be remove the phosphate group to combine a sucrose, and in this way the sucrose will be entered into the cytosol. Remember, because the UDP is using to produce the sucrose, and this is the fructose 6 phosphate will be used to attach remove the UDP to produce, and this is the sucrose will be entered into the cytosol, and UDP again used to invertase enzyme to produce fructose as well as UDP glucose. So the UDP glucose and the fructose will be formed in this way. Here you can see this fructose will be present, and hexokinase phosphoglucose isomerase enzyme used to produce fructose 6 phosphate, and this fructose 6 phosphate require the ATP, and in this way the ATP is come from the light reaction, and in this way here is the fructose 6 phosphate which that will be produced, and here is the UDP glucose, uridine diphosphate glucose containing molecule will be entered into the plastid, that is the plastid amyloplast, and here is the glucose 1 phosphate also convert through a phosphohexoisomerase enzyme to isomerization to glucose 6 phosphate again formation, and this ATP used to produce after the binding with the ADP glucose, and this glucose 6 phosphate will be remove the pyrophosphate from the ATP and itself phosphate. So this glucose 6 phosphate also convert, and this is also produced glucose 6 phosphate move into the plastid, and ADP glucose will be moved into the plastid, also the UDP glucose will be moving into the plastid. So in this way, here you can see this is the formation of the amylopectin and amylose in the amyloplast. Amyloplast is the organelle which that store the starch. So here you can see this is the glucose 6 phosphate with the AGPase enzyme used to produce the ATP, remove the pyrophosphate to produce ADP glucose. So the ADP glucose will combine with the chain of the glucose via reaction. So here is the granule bound starch synthase enzyme GBS1 which that produce the amylose, and the starch branching enzyme will produce 1 2 3 will produce the amylopectin, and in this way the three molecules glucose 6 phosphate, ADP glucose and UDP glucose will use for the combining to produce amylose and amylopectin in this sense. So I hope you make sense about these things. This is the photosynthetic video. Thank